Hi student, hi students. Myself Satish Kumar Konidala, Assistant Professor, Department of Pharmaceutical Sciences, Vignan's Foundation for Science, Technology and Research, Guntur. In this video, we are going to discuss about lipid metabolism. So we know that so many biological molecules or biomolecules are available in our body or biological system. So each and every biological molecule or biomolecule plays a major role in different types of biological reactions. So in that lipid also one type of biomolecule, it has different roles in different types of biological reactions. Okay, so let us see. So what is the importance of lipids in our biological systems, how they are playing the major role in different types of biological reactions, and what is the necessity of these lipids for daily life of biological system, and how it is going to be metabolized and excreted from our body. So let us discuss in this video. So by watching this video, student could understand the what is meant by lipid or different types of lipid molecules, and they could note the importance of lipids in our body, and they could know what is the metabolic pathways of lipids in our body, and finally they could understand the what are the associated disorders related to this lipid metabolism. Lipid metabolism. Coming to the introduction, so we know that lipids are indispensable for cell structure and function. So that means the lipid plays the major role in cell structure and function. They are utilized in the construction of cell as well as the functioning of the cell. So due to their hydrophobic in nature, so they are hydrophobic and non-polar nature. So the lipids are different than the other biological other biomolecules, and they are showing the unique action in our body. So there are different types of lipids in that like triglycerols. So so we know that lipids construct the about 15 to 20% of our body weight. So in our body weight, about 15 to 20% will be due to the presence of lipids. In that triglycerides. So triglycerides are the one type of lipid substance. So this triglyceride so they are most abundant lipids. So that means that the most type of lipids are nothing but the triglycerols, and this will comprising of 85 to 90% of our body lipids. Our body lipids. So about 85 to 90% of lipids are nothing but the triglycerols. Okay, and these lipids can construct or can comprising 15 to 20% of our body weight. Okay, and most of the triglycerols like so these triglycerols are most of the time they are stored in the adipose tissue, and they are used as the energy reserve whenever the body is required in high amounts. So whenever the body requires the high amounts of energy, then the lipids which are stored in the adipose tissue are used for the energy. Okay, so the fat also act as the insulating material. So the fat is nothing but the lipid, so they will act as the insulating material for maintaining the body temperature of our biological systems. So out of in our body weight, the lipids will comprising 15 to 20% of our body weight. In that also, most of the lipids are triglycerides. Most of the lipids are triglycerols. So these triglycerols will comprising 85 to 90% of our body lipids. Okay, and these triglycerides most of the time they are stored in the form of fat in adipose tissue, and whenever the energy is needed by our body, then the stored fat is utilized for the energy needs. Okay, and this fat is also act as the insulating material to regulate our body temperature. As I said that the triglycerols are hydrophobic and non-polar. That's why it is available, or it is stored in the form of anhydrous. So it is stored in the form of anhydrous form. Anhydrous form, that means it is without any water molecules. So the triglycerides are non-polar and hydrophobic in nature, hence they are stored in the pure form. Pure form means there is no addition of water molecule, so that they are available in the form of anhydrous form. Okay, so each triglycerides can able to produce the 9 calories. So each 1 gram of triglyceride can able to produce the 9 calories of energy. But in case of carbohydrates and proteins, they are producing only 4 calories per gram. So the reason for that one is, so in case of carbohydrates or proteins, so 1 gram of carbohydrate or protein can able to combine with 2 water molecules, 2 grams of water molecules for their storage. So for that reason, it can mix up with the water molecule, so thereby it cannot available in the pure form. So the weight of the carbohydrate or protein also including the water weight, thereby it can produce low energy when compared to the triglycerides or fat. Okay, so when there is a food deprivation or any type of fasting, so the fats can support the body energy needs. So instead of carbohydrate or proteins, so the fat will support the body's energy needs for longer periods than these carbohydrate and proteins. So for example, in case of extreme cases, human can fast and survive for about 60 to 90 days without any food. And in case of obese person, they can also survive even longer, that means for about 6 months or 1 year, so without the food. So that is the importance of fats. So they can so they can so that means they can provide the energy for a longer period, so due to their high energy production. Along with the triglycerides, other important body lipids are nothing but phospholipids, glycolipids, cholesterol. So these are the major lipid substances that are available in our body, and they can act as the components of cell membranes. And cholesterol is also a precursor for bile acids and steroid hormones. Arachidonic acid, a unsaturated fatty acid, is the substance or substrate for the synthesis of certain intracellular regulators like prostaglandins, thromboxanes, and prostacyclins. Okay, and coming to the transport of lipids, so how the lipids will be transported in our body. The insoluble lipids are solubilized in an association with proteins. So that means when they are mixed with the associated proteins, they can able to solubilize. Okay, so to form the lipoproteins. So lipids are insoluble, we know that. So lipids are non-polar and hydrophobic in nature. They are insoluble in our body fluids. So to make it soluble, we require some protein. Okay, so that means the lipids combined with the protein then can be converted into lipoproteins. Lipoproteins. So in which form? So the lipids can be transported in the blood, so for different areas of our body. So lipids are insoluble form, so they are not soluble in our body fluids. Okay, to make it solubilize, so we are require some proteins. When the lipid is combined with the protein, it will be converted to lipoprotein. So in this form, so in the form of lipoproteins, the lipids can be transported through the bloodstream for different areas of our body. Okay. Free lipids are undetectable in blood. So generally, free lipids are undetectable. That means when you go for the testing the what is the lipid content in your blood, so at that time we are not able to identify or detect the lipid content in our blood. So, but we can detect the lipoproteins. Okay, we can detect the lipoproteins. That means when the lipid is combined with the protein, that form we can able to detect, but we cannot able to detect or identify the lipids alone in our blood. Okay. Chylomicrons, so that is nothing but the very low density lipoproteins. So sorry, different types of lipoproteins are chylomicrons, very low density lipoproteins, also called as VLDL, low density lipoproteins called as LDL, high density lipoproteins HDL, and albumin free fatty acids are the different type of lipoprotein in our body. They can able to transport the lipids in the bloodstream for different areas in our body. So we know that, so lipids are insoluble in our body fluids. To transport these lipids, we require some proteins. So when the lipid is combined with these proteins, so that can be converted into lipoproteins. So here different types of lipoproteins like chylomicrons, VLDL, LDL, HDL, and albumin free fatty acids. Okay, so these are the different types of lipoproteins that can able to transport the lipid in our bloodstream to different areas of our body. So we already discussed about that. The fat or the lipids in the form of fat are stored in the adipose tissue to provide the energy for a longer period of food deprivation, that means food fasting. Okay, so in that time the fat is utilized as the energy source whenever we are in fasting. So in that case, the triglycerides, so they are already stored in the form of fat in adipose tissue. So from that the fat will be converted into glycerol and free fatty acids to produce the ATPs or energy. So how it will be happen? So this process is called as a lipolysis. Lipolysis. That means the stored lipid in the form of fat in a adipose tissue will be utilized for production of energy. So that process is called as the lipolysis. So for that reason there are some enzymes are required. So the enzyme namely hormone sensitive triglycerol lipase. Hormone sensitive triglycerol lipase is the one of the enzyme which removes the fatty acid either from carbon 1 or 3 of the triglycerol to form the diacylglycerol. The triglycerol which is stored in the form of fat in adipose tissue. So from that it will remove the triglycerol by acting or by removing the carbon 1 or 3 of the triglycerol to form the diacylglycerol. So triacylglycerol will be converted to diacylglycerol by the action of hormone sensitive triacylglycerol lipase. Okay, and other two fatty acids, so when you see in this triacylglycerol structure, so here three free fatty acids are attached to the glycerol molecule. So here this one is one, and this one, and this one. So these are the three acyl molecules or acetyl molecules are attached to the glycerol. So that's why it is called as the triacylglycerol. And one of the enzyme, that is a hormone sensitive triacylglycerol lipase. Okay, so that will first remove the either acetyl group which is attached to the carbon 1 or carbon 3. So first it will remove that one. Thereby it forms the diacylglycerol. So after that, the another enzyme, that is a lipase, which is specific for the diacylglycerol and monoacylglycerol, they can remove the remaining two acyl molecules which are attached to the glycerol molecule. So thereby we can get the glycerol and free fatty acid. So that means first, when you see in the structure of this triacylglycerol, so this is the three acetyl groups are attached to the glycerol molecule. So first, for example, here it is a 1, 2, 3. 1, 2, 3. So here the first hormone sensitive triglycerol lipase will remove the acyl group which is attached either first carbon or third carbon. So thereby it will be converted to diacylglycerol. For example, if it is remove the acyl group of first carbon, then it will be formed like this. So this is the diacylglycerol, and this will be again by the action of lipases which are specific for diacylglycerol, and so when it is removed it is formed as the monoacylglycerol. So there are two specific lipases which are act on the diacylglycerol, and another specific lipase that is act on the monoacylglycerol will remove the diacylglycerol acetyl group, and another lipase that is specific for the monoacylglycerol that will remove this acyl group which is attached in the monoacylglycerol. So thereby we will get the free glycerol and free fatty acid. So this will be utilized to produce the energy. So these are the So the removing of acyl groups from the triacylglycerol by the action of lipases is called as the lipolysis. Okay. So after forming the free fatty acids, so what is the fate of free fatty acids in our body? So that means, so after forming the free fatty acid from the triacylglycerol by the action of lipases, so what type of reactions or what type of things which are happened for the free fatty acids? So after releasing of the free fatty acid from the adipocytes or in the adipocytes, they enter into the circulation. So after forming the free fatty acids, they are entered into the blood circulation, and they are transported in bound form to the albumin. So albumin is one of the protein, so it will bound to the albumin and they are subjected to transport. And these free fatty acids enter various tissues and are utilized for the energy. So the free fatty acids which are formed from the adipose tissues, or they are released from the adipose tissues, then they are entered into the blood circulation, and there they can combine with the albumin. They are combined with the albumin, then it will be easy to transport. Okay, and during the transport they are reaching the different various tissues, or they are entering into the various tissues and utilized there. They are utilized for the energy production. So about 95% of energy obtained from the fat comes from the oxidation of fatty acids. So how it will produce the energy? When the free fatty acid, when it is subjected to oxidation, it produces the energy. Okay, so it is happened about 95%. So 95% of energy is produced from the free fatty acid is due to oxidation process. Okay, so and this oxidation process is happened in different types of tissues like different types of tissue, but it is not happened in case of brain and erythrocytes. So the oxidation process of free fatty acids to produce the energy, it is not happened in the brain and erythrocytes, but remaining type of tissues, the oxidation process is happened. Okay. So let us see the fatty acid oxidation. So oxidation process of free fatty acids. Okay, the fatty acids in the body are mostly oxidized by the beta oxidation. So we know that. So we see in that fatty acid structure. So there, alpha and beta carbons are there. So the beta carbon is subjected to oxidation. The beta oxidation may be defined as the oxidation of fatty acid on the beta carbon atom, So this results in the sequential removal of two carbon fragments, and there is a formation of acetyl-CoA. So by the removal of beta carbon atom from the fatty acids, so thereby it can able to form the acetyl-CoA. And coming to the steps and the tissues which are involved in the different stages of fatty acid oxidation. So there are three steps involved in the fatty acid oxidation. First one is the activation of fatty acid. So this will be occurred in the cytosol of cells. So we know that the fatty acid oxidation is occurred in all types of tissue except brain cells and erythrocytes. So remaining types of tissues we are seeing that fatty acid oxidation. So in that cells also, the first stage, activation of free fatty acid occurs in the cytosol of cells. And second one is the transport of fatty acid into mitochondria. So after the activation of fatty acid, it will be transported to mitochondria of the cells. Then the third step is beta oxidation proper in the mitochondrial matrix. So the beta oxidation proper is happened in the mitochondrial matrix. Okay. So the fatty acids are oxidized by the most of the tissues in our body. So it is not happened in the brain and erythrocytes. Okay. And thereby the adrenal medulla cannot utilize the fatty acid for energy requirement. So the adrenal medulla cannot be utilized the fatty acids as the energy source. Okay. So first we will discuss about the fatty acid activation. First we will see about the fatty acid activation. The fatty acids are activated to acyl-CoA by the enzyme that is the thiokinases or acyl-CoA synthetases. So for conversion of fatty acid to acyl-CoA, so that is acyl-CoA. So there are some enzymes are required. That is a thiokinases or acyl-CoA synthetases. They are called as acyl-CoA synthetases. So the three different thiokinases are required to activate the long chain fatty acids or medium chain fatty acids or short chain fatty acids. So for example, if the fatty acid is the long chain, so there is a one type of thiokinase system is required. Or if the fatty acid is a medium chain fatty acid, then another type of thiokinase system is required. Or if the fatty acid is a short chain, so the third type of thiokinase system is required. So for each type of fatty acids, there is a particular type of thiokinase system is required to convert these different types of fatty acids into acyl-CoA. Okay. So here we can see this. The fatty acid first by the action of thiokinase, it will be converted to acyl-aldenylate. So for this purpose, it will utilize the 1-ATP. So 1-ATP is required to convert the fatty acid to acyl-adenylate by the enzyme thiokinase or acyl-CoA synthetase. So during this process, 1-ATP is utilized. So after that, the acyl-adenylate will be converted to acyl-CoA by the utilization of the CoA. So that means the CoA will be attached to the acyl-adenylate. So here we can see the AMP is removed from here, and in that place we are seeing that CoA is attached. Okay. Next, transport of SL-CoA into mitochondria. So in first step, what we seen that activation of free fatty acids. So what is that? Conversion of fatty acid to the SL-CoA by the action of thiokinases or SL-CoA synthetases. Okay, and coming to the transport of SL-CoA into the mitochondria. So the inner mitochondrial membrane is impermeable to free fatty acids. So, in previous step we discussed about the activation of free fatty acid. So these free fatty acids are impermeable. So that means they are not crossing the inner mitochondrial membrane. For that reason, the free fatty acid is converted to SL-CoA. This SL-CoA is permeable for the mitochondrial membrane. So that's why the free fatty acid is converted into SL-CoA by the thiokinases. Okay, so that is already we discussed in the first step, that is activation of free fatty acid. So after that, a specialized creatine carrier system, So after converting these fatty acids into SL-CoA, a specialized carnitine carrier system, that is also called as carnitine shuttle, so which will operate the transport of activated fatty acid, this activated fatty acid from cytosol to the mitochondrial membrane, So this will occur in 4 steps. The insertion of activated free fatty acid into the mitochondrial membrane will be happening in 4 steps. In that, first one is the SL group of SL-CoA is transferred to carnitine. So SL group of SL-CoA is transferred to carnitine. There is nothing but the beta hydroxy gamma trimethyl aminobutyrate. Carnitine is nothing but beta hydroxy gamma trimethyl aminobutyrate, and this will be catalyzed by the carnitine SL transferase 1 enzyme. So that means, so we already seen this. So here we seen this SL-CoA. So this is the SL-CoA structure. So this is the SL group. So this SL group is transferred to the carnitine system, carnitine molecule. The SL group is transported to carnitine molecule. So by the action of, what is the enzyme? Carnitine SL transferase 1. So this is the enzyme which will transfer the SL group of free fatty acid to the carnitine system, carnitine SL transferase 1. So after that, the SL carnitine is transported across the membrane into the mitochondrial matrix by the specific carrier protein. Okay, and the carnitine SL transferase 2, another enzyme, the carnitine SL transferase 2, so which is found in the inner surface of inner mitochondrial membrane, and it will convert the SL carnitine to SL-CoA, Then the carnitine is released and returned to the cytosol of, so the carnitine again, so after releasing that SL group into the mitochondrial membrane, this carnitine again come back to cytosol of mitochondria, or cytosol of the cell, and again it will take the another SL group of the another free fatty acid and go to this mitochondrial membrane and transfer that SL group into the mitochondria and again come back to the cytosol. So that is the role of carnitine system. So that's why it is considered as the carnitine shuttle. So the carnitine shuttle will act as the carrier molecule to transfer the SL group of the fatty acid from the cytosol to the mitochondrial matrix. Okay. Here we can see this. So this is the cytosol. Cytosol means inside the cell. So that is the cell matrix. There we can find the carnitine cytosol transferase 1 enzyme. Carnitine SL transferase 1 enzyme, and this carnitine acyl transferase 1, it will attach the acyl group to the carnitine. It will attach acyl group to the carnitine. So this is the carnitine system. So the acyl group of acyl CoA, that is a activated fatty acid. The acyl group is attached to the carnitine. So here then it can form the acyl carnitine. And here we can see the CoA. So the CoA is remained when the acyl group is transferred to the carnitine. So only we remain CoA. And this transfer, acyl group transfer from activated fatty acid to the carnitine system is done by the action of carnitine acyl transferase 1. Whole this process is happened in the cytosol of the cell. Understood? And after transfer of acyl group to the carnitine, so here it is a mitochondria membrane. So this one is considered as the mitochondria. Okay, this is the membrane of mitochondria, and this is the inner cytosol of mitochondria. So we have seen that cell structure. So the cell consisting of a cell membrane. So inside the cell membrane there is a nucleus, and this one is considered as a cytosol. The total area is considered as a cytosol. In that cytosol, so we can see the endoplasmic reticulum, okay, and Golgi apparatus, mitochondria, and different cell organelles are there. So consider this one as a cytosol of cell. So from here we can see the free fatty acids and activated fatty acid. So in this cytosol, what will happen? The free fatty acid will be converted to activated fatty acid. So activated fatty acid is nothing but the acyl CoA. So from here, the carnitine system, the carnitine molecule will get this acyl molecule, and it will transfer to the mitochondria. This one is a mitochondria. So this is the mitochondrial membrane. The carnitine molecule will carry the acyl group of free fatty acids. Thereby it can form as the acyl carnitine, and it will transport that acyl molecule into the mitochondrial membrane. Okay, so that is the role of this carnitine shuttle. So here we seen that acyl carnitine, and it will cross the mitochondrial membrane, and it will enter this cytosol of mitochondrial matrix. There is another enzyme, that is a carnitine acyl transferase 2. So by the action of this enzyme, the acyl group of carnitine is dissociated, and there is a acyl CoA is formed. So inner side of cytosol, inner side of mitochondria, there is a CoA is there, and this acyl group, which is detached from the carnitine, will combine with the CoA by the action of carnitine acyl transferase 2. Then again it forms the acyl CoA. And here the carnitine is free here, and it will again come back to the cytosol, and it again takes the another acyl group of the fatty acid, and again it will transfer to the mitochondrial membrane. So that is happened during the transport of acyl molecule into the mitochondria. Understood? So after entering the acyl CoA into the mitochondrial membrane or mitochondrial matrix, it can subjected to beta oxidation process. Here we can see the palmitoyl CoA. So one of the fatty acid. So we can see that beta oxidation process of palmitoyl CoA. Okay. So here this palmitoyl CoA is subjected to beta oxidation. So during that process, it is undergoes 7 cycles of beta oxidation to yield 8 acetyl CoA. So one palmitoyl CoA is subjected to 7 cycles of beta oxidation to yield 8 acetyl CoA. And after yielding the 8 acetyl CoAs, each acetyl CoA can enter into the citric acid cycle. So we know that already we discussed about the citric acid cycle. So for each citric acid cycle, there is a requirement of 2 acetyl-CoAs. Okay, and this 8 acetyl-CoAs undergoes citric acid cycle and get the complete oxidized to carbon dioxide and water. So what is the end product of citric acid cycle? Liberation of carbon dioxide and water from the acetyl-CoAs. Okay, we already discussed about in the citric acid cycle. So the palmitoyl-CoA, one palmitoyl-CoA molecule is subjected to 7 cycles of beta oxidation to yield again 8 acetyl-CoAs, 8 acetyl-CoA, and this 8 acetyl-CoA will enter into the citric acid cycle. Then they are converted into carbon dioxide and water. So let us see the each cycle of beta oxidation regarding this palmitoyl-CoA. So we know that the acetyl or acyl group of fatty acid will be transferred from cytosol of the cell to mitochondrial matrix. So here we can see this acetyl-CoA is transferred from cytosol to mitochondrial matrix. So that is come into the mitochondria. So after entering this acyl-CoA into the cytosol of mitochondria, then it is subjected to beta oxidation. Beta oxidation is happened. So let us see the different steps involved in the beta oxidation. So the acyl-CoA is converted to trans-enoyl-CoA by the action of enzyme acyl-CoA dehydrogenase. Acyl-CoA dehydrogenase. The first step involved in the beta oxidation of acyl-CoA in the mitochondrial matrix is nothing but conversion of acyl-CoA to trans-enoyl-CoA. So for this process, it utilizes the 1-FAD. So thereby the FAD will be converted into FADH2. And this FADH2 will be entered into the electron transport chain. There it can produce the 2 ATPs. The FADH2, which is produced from the FAD during the conversion of acyl-CoA to trans-enoyl-CoA by the enzyme acyl-CoA dehydrogenase, it will enter into the electron transport chain and produce the 2 ATPs. Okay. After that, this trans-enoyl-CoA will be converted to hydroxy-acyl-CoA by the enzyme enoyl-CoA hydroxylase. So for this, it will require the 1 water molecule. Okay. Again, the hydroxy-acyl-CoA will be converted to keto-acyl-CoA by the action of beta-hydroxy-acyl-CoA dehydrogenase. So during this process, one NAD is utilized, and this NAD is converted into NADH, and this will be also entered into the electron transport chain. Again, it produces the 3 ATPs. Okay. And finally, the keto-acyl-CoA will be converted into again 2 acyl-CoA molecules by the action of thiokinase. Understood? So the acyl-CoA is subjected to beta oxidation. Thereby, it can able to produce the 2 acyl-CoA molecules. During this process, 5 ATPs, total 5 ATPs. One is by the 2 is by the FADH2 and 3 is by the NADH. So during in a 1 cycle of beta oxidation of acyl-CoA, total 5 ATPs are produced in different stages of beta oxidation of acyl-CoA. Finally, we will get the 2 acyl molecules in 1 cycle of acyl-CoA. Okay. But in case of palmitate acyl-CoA, so but in case of palmitate-CoA, so there are 7 beta oxidation cycles are required to convert into SL-CoA. The polymethyl-CoA, so which is having the 16 carbon atoms in their structure. So to convert this polymethyl-CoA into the SL-CoA, so it will require 7 beta oxidation cycles. 7 beta oxidation cycle. So we already discussed one beta oxidation cycle. So in one beta oxidation cycle of SL-CoA, so what will happen? 5 ATPs are produced in different stages of beta oxidation, and at the end we get the 2 SL-CoAs. Okay, and here, so there are 7 beta oxidation cycles are required to convert the polymethyl-CoA into 8 SL-CoAs. 8 SL-CoAs. Okay, so for that reason it can liberate, so during the 7 cycles it can liberate the 35 ATPs. It will liberate the 35 ATPs. And for each TCA cycle we require 2 SL-CoAs, and for each TCA cycle we will get the 18 so for each and these 8 SL-CoA molecules will enter into the each TCA cycle. So that means 8 TCA cycles are required to convert the SL-CoA into carbon dioxide and water. So for that reason it can able to produce the 96 ATPs. That means each TCA cycle can able to produce the 12 ATPs. 12 ATPs. So totally here it is a 35 ATPs and here 96 ATPs are produced during the beta oxidation of polymethyl-CoA, or the conversion of polymethyl-CoA to carbon dioxide water can able to produce the total 131 ATPs. 131 ATPs. Here we can see the energetics of palmitic acid oxidation. So we know that beta oxidation 7 cycles are required to convert the palmitic acid into SL-CoAs, 8 SL-CoAs. Okay, so during the process 7 FADH2s and 7 NADH are released. Each FADH2 can able to produce the 2 ATPs, thereby 2 into 7, thereby 2 multiplied by 7, we can get 14 ATPs. And here 7 NADH are released during the each cycle of 7 NADHs are released. That means for each cycle 1 NADH is released. So total 7 NADHs are released, and each NADH can able to produce the 3 ATPs when it is entered into the electron transport chain. So 7 multiplied by 3, then we can get the 21 ATPs from the 7 cycles of beta oxidation of this palmitic acid. And after completion of the beta oxidation, there is a 8 SL-CoAs are obtained, and these 8 SL-CoAs, when it is subjected to enter into the citric acid cycle, each SL-CoA provides 12 ATPs. Each SL-CoA provides produce 12 ATPs during the citric acid cycle to convert it into carbon dioxide and water. Okay, so then for each SL-CoA it produces the 12. So 8 multiplied by 12, it gives the 96 ATPs. So the total energies which are produced during the conversion of palmitic acid to carbon dioxide and water is nothing but 131 ATPs. 131 ATPs. Okay. And during the activation of this palmityl-CoA, so that means beta oxidation process is involved in 3 processes. First one is the activation of fatty acid, then after it is transported to mitochondrial membrane, then it is subjected to beta oxidation. Okay, so for the activation, in case of palmitic acid, there are 2 ATPs are utilized. So we are minusing the 2 ATPs. So 131 minus 2, then the net yield of ATP during the conversion of palmitic acid to carbon dioxide and water is nothing but 129 ATPs. 129 ATPs. So the bracket values are nothing but the ATPs synthesized as per P is to O ratio. P is to O ratio of 2.5 for NADH and 1.5 for FADH2. So that means how much ATPs are produced by the FADH and how much NADPH are produced. So that is a P is to O ratio is produced in bracket values. So that is about the beta oxidation of palmitic acid, and here we can see the different associated disorders related to the beta oxidation of fatty acid. So first one is a SIDS, SIDS. So this is the one of the disorder due to the blockade of beta oxidation. So when the beta oxidation is prevented, that may lead to the development of SIDS disorder. So this SIDS disorder is the sudden infant death syndrome. SIDS is nothing but sudden infant death syndrome. So it is an unexpected death of healthy infant, unusually overnight. So it is generally happened in overnight. The real cause for this SIDS is not known, but it is now estimated that at least 10% of SIDS is due to the deficiency of medium chain acyl CoA dehydrogenase. I already explained that for each type of fatty acid, that is for long chain fatty acid, medium chain fatty acid, and short chain fatty acids, different type of enzyme system is required. Different types of enzyme system is functions. Okay. So in case of this SIDS syndrome, the deficiency of medium chain acyl CoA dehydrogenase is the reason. So out of 100, 10 SIDS are due to the deficiency of medium chain acyl CoA dehydrogenase. So this enzyme defect has an frequency of, so for example, 1000 babies are born, 10,000 births are done. So in that, one baby may got this deficiency of medium chain acyl CoA dehydrogenase. Okay. So this is more prevalent than the phenylketonuria. So this disease is more prevalent than the phenylketonuria. The occurrence of SIDS is explained as follows. So we can see how the SIDS can occur. So glucose is the most important source of energy soon after eating or feeding the babies. So when the babies eat or feed the milk from the mother, the glucose is the principal source of energy. Okay. So after few hours of this feeding, the glucose level as well as the utilization is decreased. So why? Because the feeding is happened. So after the feeding, so there is no glucose is present in the blood of babies. So thereby the glucose level as well as the utilization of this glucose is decreased. So then what is another source of energy? The fatty acid is the another source of energy. So the beta oxidation of fatty acids will come into the picture, and it can serves the energy need. It can serve the energy need. So during that process, when there is a beta oxidation is blockade is occurred. How it will occur beta oxidation from blockade? By the lack of medium chain acyl CoA dehydrogenase. So what is the enzyme role here? We already seen different steps involved in the beta oxidation of fatty acid. So what is the first step? So the acyl CoA will be converted into trans-enoyl CoA. Acyl CoA is converted into trans-enoyl CoA by the action of acyl CoA dehydrogenase. So in case of babies, medium chain fatty acids are there, that will be converted into trans-enoyl CoA by the enzyme medium chain acyl CoA dehydrogenase. When this medium chain acyl CoA dehydrogenase deficiency occurs in the babies, then the beta oxidation will be blocked. So thereby the energy is not produced to the baby, and that means the glucose is not available or energy is not produced, thereby it may lead to sudden deaths. So that is a happen in case of SIDS. So this SIDS disorder is occurred only one baby out of 10,000 babies birth. Okay. Next one is a Jamaican wanting sickness. Jamaican wanting sickness. So this disease is characterized by the severe hypoglycemia. Hypoglycemia is low levels of glucose in our blood and vomiting, convulsions, coma and death. These are the some symptoms of Jamaican wanting sickness, and it is caused by the eating of unripe ackee food. Ackee fruit. So when you eat the unripe ackee fruit, so that may lead to blockade of this beta oxidation of fatty acids, thereby it leads to hypoglycemia, vomiting, convulsions, coma and death. Okay, so what is the reason for that? The reason is nothing but the unripe ackee fruit consisting of a toxic amino acid, that is nothing but hypoglycin A. So the unusual toxic amino acid which is present in the unripe ackee fruit is nothing but hypoglycin A. So this toxic amino acid will inhibit the enzyme acyl-CoA dehydrogenase. Again, the first step of beta oxidation of the fatty acid is nothing but conversion of acyl-CoA to trans-enol-CoA. So by the action of dehydrogenase, acyl-CoA dehydrogenase. When this enzyme is blocked by the hypoglycin A, then the total oxidation process is blocked, thereby the energy is not produced. So that is another disorder of beta oxidation blockage for the fatty acids. Okay, these are the two disorders associated with the beta oxidation of fatty acids. In next class we are going to discuss about the biosynthesis of fatty acids. Thank you.